All right, we'll move on to the next talk, and that's volatile organic analysis of return lunar samples by Danielle Simkus.
So my name is Danielle, and I work in the Astrobiology Analytical Lab at NASA Goddard. And today I'm going to talk about organic volatiles specifically and the relevant analyses that could be performed on returned lunar samples. Next slide. So my research group focuses on analyses of organic compounds that are of specific interest for astrobiology. In terms of target molecules, my research group often focuses on analyses of amino acids specifically because these are the chemical building blocks of protein in terrestrial biology and are essential to life on Earth. And we want to know how these compounds are formed by non-biological reactions in space in order to understand how common these prebiotic molecules are in the solar system. But we're also very interested in analyzing the precursors to amino acids, the precursors to amino acids, including things like cyanide, amines, carboxylic acids, aldehydes, ketones. All of these volatiles have been detected in carbonaceous chondrite meteorites, which are the most common type of sample that we analyze. And amino acids and cyanide species have been detected previously in some lunar samples, and we're interested to fill in the gaps and find out exactly where these compounds are coming from. Next slide. So the lab methods that we use cannot be carried out in situ on the lunar surface. We really do need return samples in order to extract, process, and analyze these molecules in the lab. And from one single lunar sample, we're able to extract, isolate, separate, detect individual amino acids and volatile organics using these compound-specific techniques, such as liquid chromatography mass spectrometry and gas chromatography mass spectrometry. We also have a method to analyze insoluble or bound cyanide within the residual solid after the hot water extraction. So for the amino acid analyses specifically, we're not only detecting what we call free or water-soluble amino acids, we're also subjecting a portion of our samples to acid hydrolysis in order to release amino acids that may have been bound within larger polymers, and to synthesize amino acids from smaller volatile precursors that are present in the samples. So for these reasons, we want to know exactly what those volatile precursors might be within the samples in order to get a sense of how amino acids are synthesized, not only during the sample workup, but as well as in the space surface. Next slide. So not to go into too much detail here, but I just wanted to highlight some of the mechanisms by which volatile organics could react and potentially yield amino acids. And here we've highlighted some mechanisms in orange to indicate certain reactions that could take place naturally in space, and others that we may expect to take place on Earth during lab workup. And measuring the relative abundances of each of these compound classes can give us an indication of what types of reactions may be playing more significant roles. So our overarching questions for soluble organic analyses of lunar samples are essentially, what is the distribution of amino acids and volatile organics on the lunar surface? And where did they come from? In addition to any chemical reactions that we might take place on the lunar surface, there are three potential external sources of volatile organics that we're considering. Could you go to the next slide? So these external sources that we're considering are meteoritic or cometary delivery, implantation of carbon by solar wind, and also terrestrial contamination. And we're interested to know how significant are each of these potential sources of volatile organics, and what processes have the greatest influence on determining the distribution of organics on the lunar surface. Could it be these external sources? Is it temperature, or is it destructive processes such as evaporation and degradation under UV? Next slide. So if one of the major influences on the presence of volatile organics is the concentration of these compounds via cold trapping, we would expect to see relatively higher concentrations of both amino acids and volatile organics at the poles and within the shadowed regions on the lunar surface. And then we'd expect to see relatively lower abundances within the sunlit and unshadowed regions. And I should note that this cartoon that we're showing here and that will be on the following two slides are not completely accurate for all shadowed regions on the lunar surface. Of course, most surfaces will be shadowed without actually being sheltered underneath a boulder, but these images were created for a specific project where this was actually the case. Just clarifying this so that it doesn't cause any confusion. Next slide. So in the case that the delivery of organics through meteorite impacts and solar wind implantation occurs at a faster rate than the destruction or loss of these molecules through radiation damage or evaporation, we would expect to see the highest abundances of amino acids and the volatile precursors in exposed areas and at the lunar surface, and then a decrease in abundance with increasing depth. Next slide. And in terms of the role of UV radiation, we can use the relative abundance of amino acids versus volatile organic compounds as indicators for whether UV serves as a destructive force or whether it's involved in the synthesis of amino acids. In the former case, we would expect higher ratios of amino acids to volatiles in areas that are shielded from UV, for example, in the shadowed regions or at the greater depths. In the latter case, we'd expect higher ratios in areas that are exposed to UV. In the latter case, we would expect higher ratios in areas that are exposed to UV or at the shallower depths in the regolith. Okay, next slide. So in conclusion, in terms of sample requirements, what we really need is the ability to compare and contrast samples from distinct environments. For example, shadowed regions versus unshadowed regions, or surface samples versus subsurface samples. And most importantly, we need to know that the collection and the curation of the lunar samples have both been carried out with volatile organics in mind.
3 minutes left.
Okay. Tomorrow afternoon, my postdoc advisor, Jamie Elsela, will be going into quite a bit more detail about curation requirements and the history of organic analyses of lunar samples. So we can discuss this in more detail then. But over time, we are definitely moving in the right direction in terms of optimizing the collection and curation of samples. For the initial analyses of Apollo samples, although these studies did provide some of our first insights into the organic content of lunar samples, there were definitely some unknowns in terms of curation conditions and potential sources of contamination. With ANCSA, we're very lucky to have specially curated samples from a range of environments, but even with special curation, we still don't fully understand how 50 years of storage could potentially affect the preservation of volatiles. So with Artemis, we can essentially have the full package and have fresh samples, proper curation, and a range of new and contrasting environments. So my lab group is definitely excited to be a part of these discussions and planning. And with that, I will leave this slide up in case there are any questions.
Open for discussion. Okay, if there isn't any, I actually have one. With your analytical techniques, can you do measurements of enantiomeric excesses? Yeah, we can. I mean, yes. So enantiomeric excesses are actually one of our key interests, at least for amino acids. Some of our techniques for other volatile organics allow us to look for enantiomeric excesses as well. And also in any case where we have enough sample, we could also do stable isotope analyses, generally carbon isotopes. So these are two techniques that might be able to give us some indication of contamination versus extraterrestrial sources. Great. Thank you. Now, in the interest of time, I'm going to hand it over to the co-chair, and she's going to run the session that's focused on upcoming missions.